today I'm going to be talking to you a little bit about the role of T cells in bluetongue virus infection in sheep, which is the current project that I'm working on. So as many of you know, bluetongue virus, or BTV, is the causative agent of bluetongue. It's an infectious, non-contagious hemorrhagic disease of ruminants, and it causes particularly severe clinical disease in sheep. As you can see, it causes facial edema, inflammation of the coronary band, and reddening of the eyes and mucosal membranes. It's arthropod-borne, so it's transmitted through the bite of an infected Culicoides midge. And currently, whilst vaccination of susceptible livestock is at the cornerstone of successful BTV control, the problem that we have is that the monovalent inactivated vaccines that are available don't provide cross-protective immunity against the many heterologous BTV serotypes, of which there are at least 27. So really we want to try and understand more about the host response to BTV to try and hopefully eventually develop more novel cross-protective vaccines. And so we know that neutralising antibodies do provide protection, but the level to which they're cross-protective we're not quite sure yet. And so our question is basically whether T lymphocytes could provide this cross-protective immunity, particularly as both CD4 and CD8 positive T cells have both been known to recognise epitopes on BTV structural and non-structural proteins. And aside from this, it's also important from the point of view of pathogenesis, particularly their role in viral replication and also at the site of infection within the local skin environment. So a recent in vivo study carried out at the Pirbright Institute looked at the role of T cells, in BTV1 infected calves. And we found that CD4 depleted calves had higher clinical scores, impaired viral clearance, and also higher BTV RNA levels, whereas the WC1 gamma delta T cell depleted calves were found to have an earlier detectable increase in BTV RNA within the blood at 1 to 4 days post-infection. The major caveat, though, to this study was that the peak viremia, which in cattle is typically 8 to 12 days, poorly overlapped that of the T cell depletion window, as you can see at the bottom. So really the project that I'm working on hopes to kind of almost replicate this in sheep, to look at the role of T cells on clinical disease, viral clearance, antiviral responses, and also transmission to and from the Culicoides biting midges, but in BTV-infected sheep. And hopefully, because the viremia is earlier, it should overlap better with the T cell depletion window. So to do this, we intravenously inoculate monoclonal antibodies into the sheep to deplete either CD4 positive T cells, CD8 positive T cells, WC1 positive gamma delta T cells, and then we also have an isotype matched antibody that we inject into a separate group as a control for these injections. We then use a natural in vivo infection model where we infect adult female Culicoides sonorensis and basically allow them to feed on a blood meal containing BTV4, which is our serotype of choice. And then once the extrinsic incubation period has happened and you get replication and dissemination of the virus through the midge, we then allow them to feed on the sheep to establish infection. So overall, we're hoping to have 7 sheep in every group. So far, the numbers, the data that I'm going to talk about today is based on these numbers here, which correspond to the first 3 animal experiments that we've performed. And in every replica of the animal experiment, we have 1 non-depleted, non-infected control sheep, which basically acts as a true control and also to monitor for contact transmission as well. We chose BTV4 as well because it's a moderate strain, so it allows us to detect changes above and below the clinical severity of the normal strain, and also because it has a high vector competence, so it allows us to actually do this blood feeding naturally rather than having to intrathoracically inoculate the midges. So we're going to look at the dynamics of immune cell populations in the blood, so both T cells and B cells, the T cell depletion dynamics, the clinical signs and pathology at post-mortem, the virus production through titration and PCR, the transmission to and from Culicoides midges, and then 3 things I'm not going to talk about today are the skin environment, the serology, and also the T cell responses as well. So in order to monitor the depletion of different T cells, I use flow cytometry and basically label the sheep blood at various time points throughout the course of the infection for different immune cell markers. So you can see on the left, there's the CD4, on the y-axis is the CD4 positive cells, and you can see there's about 15% in that particular sheep before infection and depletion. In the middle panel, you can see on the y-axis is the number of positive CD8 positive cells for the CD8 marker, and you can see in the sheep was about 5%, and then again in different sheep, WC1 positive gamma delta T cells is about 7%, and this is based on two different markers, the WC1 marker and also the gamma delta T cell receptor marker, which is on the x-axis. So when we look at 2 days post-infection, which is following infection and depletion, you can see that basically the numbers have drastically dropped and the depletions worked quite well. So there's 0.53% of CD4 positive cells left, 0.257% of CD8 positive T cells left, and then only 0.008% of WC1 positive gamma delta T cells left. So the depletion seems to work quite nicely. So when we look at this over the course of infection, so you can see in the left-hand top panel, you can see the number of CD4 positive T lymphocytes per ml of blood. The right-hand panel is the CD8 T cells, and the bottom panel is the WC1 positive gamma delta T cells. And so compared to the control, which is the black line that kind of stays quite stable throughout the course of infection, you can see that the depletion happens quite nicely, and the depletion is between -2 to 2 days post-infection that we have these injections of monoclonal antibodies. You can see that whilst on the bottom panel, the WC1 positive gamma delta T cells do stay quite nicely down for the whole duration of the experiment. The CD4 and the CD8 positive T cells do start to come back, which we've seen before in other experiments. And we believe that this might be due to incomplete depletion within the lymphatic organs, which is why they do start to come back when we stop the injections. So then when we look at the levels of BTV viremia and the dynamics across the course of the infection, you can see plotted on the y-axis is the CT value, which is based on a BTV segment 10 PCR. So you can see basically the average of all the animals that are in each particular group. So you can see that the CD4 positive T cell depletion group appears to have an earlier detectable increase in BTV viremia within the blood, and also a higher overall level compared to the isotype control, which is in red. You can see that the blue line, which is the WC1 positive depletion group, appears to have a kind of later peak viremia. However, this is actually just based on one of the animals, because the other ones, based on the error bars, basically didn't show the same pattern. So this looks like it might be animal-specific variation at the moment, but we can see when we have the final few replicates if this holds up. And then also, I guess at the end of the graph, you can see that the CD8 and WC1 depleted groups appear to have a higher viremia later on in infection, which might suggest that the CD8 and the WC1 positive T cells do have a greater role in viral clearance, because when they're not there, you get more virus still remaining in the sheep. And obviously the control stayed negative throughout the experiment, which suggests that there was no contact transmission between the animals. So when we look at clinical severity between each of the depletion groups, basically each line is a different animal, and you can see from the bottom panel, the isotype control, that there's quite a varied range of clinical severity between the animals. There's a lot of animal variation, but overall it does appear to be, as we expected, a moderate strain with some variation. So when you compare this to the WC1 depleted groups and the CD4 depleted groups, you can see that the clinical severity does seem to increase quite a lot. We still have more replicates to do, so we're hoping more maybe that this holds up, we're not sure. But so far it looks like the depletion of these two T cell groups do appear to increase the severity of clinical disease. Interestingly, you can see that the CD8 depletion group appears to cause reduced symptoms, so they're a lot milder compared to the isotype control. So again, this will be quite interesting to see if this holds up. And based on body temperature, so this is body temperature on the y-axis plotted against days post-infection, you can see that whilst the isotype control, the CD4 and also WC1 groups appeared to reach high fever at peak viremia, the CD4 group didn't ever really reach that based on averages of the animals so far. So again, this does appear to suggest milder clinical symptoms when the CD8 cells aren't there anymore. And then finally, we also wanted to look at transmission from the host back to the midges. So basically, we fed uninfected Collocoides sonorensis midges on the B2 infected sheep at their peak viremia, and the average peak viremia of each of the sheep for each group are given in the blue bar at the bottom. And so essentially, we took, following feeding, we took 8 to 16 midges and processed these homogenised them, performed RNA extraction and BTV segment 10 PCR, basically to determine the overall viral uptake within these midges. So the average is shown in the black bars here. And then essentially we collected the others, incubated them for 8 days to allow extrinsic incubation within the midge and replication. And so when we then process these in an identical manner, anything that we then plot on the graph that's above the black bars suggests that there's some form of viral replication happened within the midge. And then we've used, for fully transmissible infection, we've kind of used a conservative cutoff of about 25 CT. We kind of need to do more work on this to figure out if this is the true reflection, but that's based on previous published data. So when we then plot the incubated midges, interestingly, you can see in the isotype control group, there's about 46 midges which had a fully transmissible infection based on our cutoff. But when you compare this to the CD8 group, which has an equivalent viremia in the sheep, so they were feeding on sheep that had the same kind of viremia, there's almost double the amount of midges that have a fully transmissible infection. Similar to the WC1 group, they have 64, which is again a lot higher than the isotype control. And then when you look at the CD4 group, they actually have more similar to the isotype control, if not slightly less. Again, we need to continue processing all of the replicates, but so far it looks quite interesting. It may suggest that the depletion of the CD8 and the WC1 gamma delta T cells may increase transmission of BTV at peak viremia in these sheep. So in conclusion, our natural in vivo infection model resulted in successful transmission of BTV to all 16 sheep, and we got typical clinical signs of high fever, edema and hemorrhage. The WC1 gamma delta T cell depletion was successful, and we got significant depletion of CD4 and CD8, although these did come back towards the end of infection, and we think this might be because of incomplete depletion of the lymphatic organs. We got significant variation in clinical disease manifestation between the animals. However, overall we found so far that the CD4 and the WC1 gamma delta T cell depleted group appears to increase severity. However, the CD8 depletion tends to result in milder clinical signs and reduced fever. The viremia dynamics, so the CD4 depletion resulted in earlier detectable increase and higher overall levels of BTV in the blood, similar to the cattle. The CD8 positive and the WC1 positive gamma delta T cells may have a greater role in BTV clearance. And then finally, the CD8 and the WC1 gamma delta T cell depletion appears to result in increased transmission of BTV to Calicoides sonorensis at peak viremia. So with that, I'd like to thank the Orbovirus Research Group, particularly Karen, Mark, Lindsay and Karen for their help with processing all the samples, Katie and Simon, the entomology group for all of their help processing the samples, the REF lab for all of their help, and also Animal Services. Thank you.
Thank you.